Phytoplasmas are a specific group of phytopathogens that fill the gap between bacteria and viruses. Despite the fact that phytoplasmas were discovered only in 1967, symptoms of disease had been known long, and they are quite different. Plant dwarfism, fruit deformation, witch's broom, shoots dieback, phyllody, etc. This is not a complete list of possible symptoms. Such a wide symptom range does not allow to identify visually the infection with an accuracy of 100%, as the nature of these changes can be caused by virus infections or physiological damages. The problems are that not always do phytoplasmas show symptoms. They can be in latent period. What does that mean? Visually, the plant is healthy, but when conducting some tests, specialists reveal a high concentration of phytoplasma. Nowadays, there are over 100 identified Phytoplasma species. Some of them belong to quarantine pests in many countries, such as grapevine flavescens, Doré Phytoplasma, which is still absent in the Russian Federation. This great pathogen is spread in Europe, in countries like France, Italy, Portugal, and Spain. Every year, Phytoplasma spreading is increasing. It enters the viticulture zones of Eastern Europe. Phytoplasma vectors are various Cicadelidae and Psyllidae species. The infection can harbor in weeds such as dodders, nettle, and bindweed. Phytoplasmas cause great economic loss to agriculture. For instance, phytoplasma causing blackwood of grapevine in places of the infection spreading leads to the loss of up to a third part of the yield. And when the infestation reaches 70% of the whole number of vines, vineyards are completely uprooted. Such phytoplasma species as grapevine flavescens Doré Phytoplasma are already considered to be highly dangerous and are included in quarantine lists of many countries. With no protection measures in grapevine flavescens Doré outbreaks, the plant gets infested up to 100% during several years. It means plantations get infested 100%. The control is difficult because the phytopathogen is an intracellular parasite. Controlling phytoplasmas is handicapped by their intracellular parasite nature, and curing infested cells does not bring tangible results. Besides, the pest overwinters in plant roots, and in spring and in summer, it gets back to the above-ground organs with sap concentrating in phloem tissue of young shoots and leaves. It means that while monitoring, young shoots and leaves should be sampled with leaf stalks included. If these peculiarities are not taken into account, the pathogen can be detected late and spread to pest-free areas. It is possible to prevent the spreading either by treating plants for planting with antibiotics which are not allowed to be used in the Russian Federation, or heat treatment. Unlike bacteria, these microorganisms do not cultivate and cannot be tested by traditional methods. Precise phytoplasma diagnostics became possible with molecular methods development, with researchers detecting phytopathogen DNA fragments from plants. The specialists of Research and Testing Department at All Russian Plant Quarantine Center developed methods of phytoplasma diagnostics using PCR and sequencing. We can identify phytoplasma with an accuracy of 100% using these two methods. It includes species identification. I suppose such studies can help us to expand the knowledge about phytoplasmas, as well as use the developed methods for phytosanitary testing. To detect and identify the causing agent of grapevine flavescens Doré and apple proliferation, there have been developed and enhanced methods of PCR. Analysis of the restriction fragments length polymorphism RFLP, as well as sequencing. Besides, specialists have detected phytoplasmas causing stolbur of potato, beetroot, and raspberry, pear decline, strawberry phyllody, and blackwood of grapevine. The results of FGBU Viniker's scientific and research work on phytoplasma diagnostics in the conditions of import substitution can be successfully applied to maintain the phytosanitary security in Russia.